Hello, everyone. How are you doing? Now that we have gone through the whole topic, I just want you to see illustrations on how neurotransmitters work. Just as a reminder, neurotransmitters are chemical messengers that transmit signals across synapses, enabling neurons to communicate effectively with one another. They bind to specific receptors on postsynaptic neurons, thereby triggering various responses. We talked about dopamine, serotonin, and all of them. Dopamine is highly involved in reward and motivation, while serotonin is very essential in mood regulation and appetite regulation. So an imbalance in any one of them would lead to disorders like depression. For example, depression has been linked to serotonin and dopamine imbalance, and Parkinson's disease has been linked to dopamine deficiency. Okay, so other neurotransmitters that we've talked about include acetylcholine, which is very essential in learning and memory, your gamma aminobutyric acid, and glycine. They are inhibitory neurotransmitters. Thereby, they enhance relaxation. Remember your glutamate, it is an excitatory neurotransmitter, highly essential in learning. So when we understand each of their mode of action and their medical implication, it is highly essential for therapy. For example, the medical significance has led to treatments for depression and also dopamine replacement therapy for Parkinson's disease. So I want you to enjoy the video and for better understanding. Best of luck.
Communication between neurons and communication between neurons and muscle occurs at specialized junctions called synapses. The most common type of synapse is the chemical synapse. Here, we examine the events that take place at the neuromuscular junction, a chemical synapse using the neurotransmitter acetylcholine. Synaptic transmission begins when the nerve impulse reaches the presynaptic axon terminal. Depolarization of the presynaptic membrane initiates the sequence of events leading to transmitter release and activation of receptors on the postsynaptic membrane. Shown here are the principal molecules and organelles necessary for release of neurotransmitter. Synaptic vesicles exist in distinct pools, either tethered to the cytoskeleton in a reserve pool or free in the cytoplasm. Some of the free vesicles make their way to the plasma membrane and dock as a series of priming reactions prepares the vesicular and plasma membranes for fusion. The membranes of the synaptic vesicles are drawn together via protein complexes, collectively called snares, that are expressed on the vesicle and presynaptic membrane. When the axon terminal is depolarized, voltage-gated calcium channels open, and calcium ions rush into the axon terminal. Some of the calcium ions bind to a protein on the synaptic vesicle membrane called synaptotagmin. When calcium binds to synaptotagmin on the synaptic vesicles nearest the active zone, the vesicles are drawn even closer to the presynaptic membrane. The vesicles fuse with the axon terminal membrane and release their transmitter cargo into the synaptic cleft. Some of the transmitter molecules bind to special receptor molecules in the postsynaptic membrane. The response of the postsynaptic cell, either excitation or inhibition, depends upon the particular neurotransmitter and receptor combination. For example, the receptor for acetylcholine is permeable to sodium. After binding acetylcholine, the channel opens and sodium ions enter the postsynaptic cell, thereby generating an excitatory postsynaptic response. Transmitters are inactivated or removed rapidly from the synaptic cleft so that transmission is brief and accurately follows the presynaptic input signal. For acetylcholine, an enzyme in the synaptic cleft, acetylcholinesterase, breaks down acetylcholine into choline and acetate. The release of transmitter from the receptors causes the channels to close. Not all transmitters are broken down by enzymes in the synaptic cleft. Many transmitters are rapidly cleared from the synaptic cleft by being taken up into the presynaptic terminal by special proteins called transporters. This process is known as reuptake. Reuptake not only cuts off the synaptic activity promptly, but also allows the terminal to recycle transmitter molecules. The membrane needed for creating synaptic vesicles is also recycled via endocytosis of the presynaptic membrane. The recycled vesicles are refilled with neurotransmitter molecules and are ready for another round of synaptic transmission. Acetylcholine, abbreviated ACH, was the first neurotransmitter to be identified. Acetylcholine is synthesized in the nerve terminal from acetyl coenzyme A and choline in a reaction catalyzed by the enzyme choline acetyltransferase. Neurons that use acetylcholine as a transmitter are called cholinergic neurons. After release, acetylcholine is rapidly metabolized by the enzyme acetylcholinesterase. Acetylcholinesterase breaks down acetylcholine into choline and acetate. The choline is transported back into the nerve terminal by membrane transporters and used to resynthesize the neurotransmitter. In the brain, several distinct clusters of cholinergic cells are present. The basal forebrain includes major groups of cholinergic cells in the medial septal nucleus, the nucleus of the diagonal band, and the nucleus basalis. These cholinergic cells project to the hippocampus and amygdala, as well as throughout the cerebral cortex. A smaller group of cholinergic cells in the pedunculopontine and laterodorsal tegmental nuclei project to the reticular formation and thalamus and are believed to be involved in arousal and the sleep-wake cycle. Cholinergic systems in the cortex are crucial for learning and memory, and widespread loss of these cells is a characteristic of Alzheimer's disease. Acetylcholine also acts at a variety of postsynaptic targets in the peripheral nervous system, preeminently at the neuromuscular junction of striated muscles and in the visceral motor system. There are two broad classes of ACH receptors, nicotinic and muscarinic receptors. These receptors are so named because of the drugs nicotine and muscarine that selectively activate each of the receptors. Most nicotinic receptors are ionotropic, responding rapidly and usually having an excitatory effect. The drug curare, once used as an arrow poison by South American Indians, blocks nicotinic ACH receptors. Because the synapses between nerves and skeletal muscles are nicotinic, curare paralyzes all skeletal muscles, including those used in breathing. Muscarinic receptors are G-protein coupled, metabotropic receptors, so they have slower responses when activated, and they can be either excitatory or inhibitory. Muscarinic receptors can be blocked by the drugs atropine or scopolamine. Certain organophosphate insecticides, such as diphenyl trichloroethane, DDT, and the nerve gas sarin, inhibit the enzyme acetylcholinesterase, causing ACH to accumulate at the synapse. This buildup of acetylcholine depolarizes the postsynaptic cell and renders it refractory to subsequent acetylcholine release, causing, among other effects, neuromuscular paralysis. Many other drugs also act at cholinergic synapses. For example, botulinum toxin is a potent bacterial poison that can grow in improperly canned food. Botulinum blocks the release of acetylcholine, resulting in muscular paralysis and asphyxiation. In much diluted form, botulinum toxin is marketed as Botox, which inhibits facial wrinkling by locally paralyzing facial muscles. Tetanus toxin is chemically similar to botulinum and is responsible for tetanus poisoning, lockjaw. Finally, black widow spider venom causes the continuous release of acetylcholine at the neuromuscular junction, causing the muscle to respond violently until the supply of acetylcholine is depleted.
The amino acid glutamate is the principal excitatory neurotransmitter in the brain, and it is estimated that over half of all brain synapses release this agent. Like all amino acids, glutamate has both a carboxyl and an amino functional group attached to the same carbon. Also attached to this carbon is a side chain. Glutamate is a non-essential amino acid that does not cross the blood-brain barrier and must therefore be synthesized in neurons from local precursors. The most prevalent precursor for glutamate synthesis is glutamine, which is released by glial cells. Once released, glutamine is taken up into presynaptic terminals and metabolized to glutamate by the mitochondrial enzyme glutaminase. Glutamate can also be synthesized by transamination of 2-oxoglutarate, an intermediate of the tricarboxylic acid cycle. Hence, some of the glucose metabolized by neurons can also be used for glutamate synthesis. Glutamate is packaged into synaptic vesicles by transporters termed V-glute. At least 3 different V-glute genes have been identified. Once released, glutamate binds to glutamate receptors on the postsynaptic cell. The glutamate is removed from the synaptic cleft by the excitatory amino acid transporters. There are 5 different types of high-affinity glutamate transporters, some of which are present in glial cells and others in presynaptic terminals. Glutamate taken up by glial cells is converted into glutamine by the enzyme glutamine synthetase. Glutamine is then transported out of the glial cells and into nerve terminals. In this way, synaptic terminals cooperate with glial cells to maintain an adequate supply of the neurotransmitter. This overall sequence of events is referred to as the glutamate-glutamine cycle. Several types of glutamate receptor subtypes have been identified. 3 of these are ionotropic receptors called, respectively, NMDA, AMPA, and kainate receptors. These glutamate receptors are named after the agonists that activate them: N-methyl D-aspartate, alpha-amino 3-hydroxyl 5-methyl 4-isoxazole propionate, and kainic acid. All of the ionotropic glutamate receptors are non-selective cation channels, allowing the passage of sodium and potassium ions and, in some cases, small amounts of calcium. Hence, AMPA, kainate, and NMDA receptor activation always produces excitatory postsynaptic responses. Among the many interesting properties of NMDA receptors, perhaps the most significant is the fact that the ion channel allows the entry of calcium ions in addition to monovalent cations such as sodium and potassium. As a result, EPSPs produced by NMDA receptors can increase the intracellular concentration of calcium. This intracellular calcium can then act as a second messenger to activate intracellular signaling cascades. Another key property is that they bind extracellular magnesium. At hyperpolarized membrane potentials, this ion blocks the pore of the NMDA receptor channel. Depolarization, however, pushes magnesium out of the pore, allowing other cations to flow through. Thus, NMDA receptors pass cations, most notably calcium, only during depolarization of the postsynaptic cell, a property that is the basis for some forms of synaptic plasticity. Another unusual property of NMDA receptors is that the opening of the channel of this receptor requires the presence of the obligatory coagonist, the amino acid glycine. Thus, both the glutamate and glycine sites must be occupied for the activation of the receptor. In addition to these ionotropic glutamate receptors, there are 3 types of metabotropic glutamate receptor. These receptors, which modulate postsynaptic ion channels indirectly, differ in their coupling to intracellular signal transduction pathways and in their sensitivity to pharmacological agents. Metabotropic receptors activate G proteins, which then dissociate from the receptor and interact directly with ion channels, or bind to other effector proteins such as enzymes that make intracellular messengers that open or close ion channels. Metabotropic receptors cause slower postsynaptic responses than ionotropic receptors, and the physiological roles of metabotropic receptors are therefore quite varied.
The catecholamines, norepinephrine, epinephrine, and dopamine, belong to a wider group of neurotransmitters called monoamines, that is, compounds possessing a single amine group. All of the catecholamine transmitters are synthesized from the amino acid tyrosine in a succession of metabolic steps, each controlled by a different enzyme. The presence or absence of the successive enzymes determines which transmitter is synthesized by a given neuron. In a reaction catalyzed by tyrosine hydroxylase, a hydroxyl group is added to tyrosine, yielding L3,4-dihydroxyphenylalanine, L-DOPA. Tyrosine hydroxylase is the rate-limiting enzyme in catecholamine synthesis. A second enzyme, aromatic L-amino acid decarboxylase, catalyzes the removal of a carboxyl group to yield dopamine. In noradrenergic neurons, the enzyme dopamine beta hydroxylase catalyzes the conversion of dopamine into norepinephrine. This conversion occurs within the synaptic vesicle. Neurons in the brain that release norepinephrine, abbreviated NE, are located throughout the brainstem, most notably in the locus coeruleus. Because norepinephrine is also known as noradrenaline, norepinephrine-producing cells are said to be noradrenergic. The output of the noradrenergic locus coeruleus cells extends broadly throughout the cerebrum, including the cerebral cortex and the thalamic nuclei. In addition, the output of these cells projects prominently to the cerebellum, pons, and spinal cord. Because of this wide distribution of projection paths, noradrenergic cells of the locus coeruleus are believed to modulate many behavioral and physiological processes, including mood, overall arousal, attention, and sexual behavior. Like dopamine, norepinephrine may coexist in the same nerve terminal with other neurotransmitters and modulators such as enkephalin, vasopressin, or neuropeptide Y. Norepinephrine is cleared from the synaptic cleft by the norepinephrine transporter, which is also capable of taking up dopamine. The CNS contains 4 subtypes of norepinephrine receptors: alpha-1, alpha-2, beta-1, and beta-2 adrenergic receptors, all of which are metabotropic receptors. They're usually referred to as adrenergic receptors because they're sensitive to both norepinephrine and epinephrine. Like dopamine, norepinephrine is degraded by MAO and catechol-O methyltransferase. Many drugs that affect dopamine synapses also affect noradrenergic synapses. For example, reserpine prevents the storage of monoamines in synaptic vesicles. The norepinephrine transporter serves as a molecular target of cocaine and amphetamines, which act as stimulants by producing a net increase in the release of norepinephrine and dopamine. Drugs have also been developed that are specific to norepinephrine receptor subtypes. For example, the so-called beta blockers are used to regulate blood pressure and autonomic function. The indoleamine serotonin is synthesized from the amino acid tryptophan in two chemical steps. The enzyme tryptophan hydroxylase adds a hydroxyl group to the amino acid to form 5-hydroxytryptophan. The enzyme aromatic L amino acid decarboxylase then removes a carboxyl group from the molecule to yield 5-hydroxytryptamine. Because its chemical name is 5-hydroxytryptamine, serotonin is abbreviated 5-HT. Serotonergic cell bodies are relatively few and are concentrated in the Raphe nuclei of the midbrain and brainstem along the midline. Large areas of the brain are innervated by serotonergic fibers. Serotonin has been implicated in the control of sleep and wakefulness, mood, anxiety, and many other functions. Termination of the effects of synaptically released serotonin occurs largely by means of a reuptake process. Cytoplasmic serotonin is also metabolized by the enzyme monoamine oxidase, or MAO. A large number of serotonin receptors have been identified. All but one are metabotropic receptors. A number of drugs have been found that stimulate 5-HT release, for example, fenfluramine and many amphetamines, and thus act as potent agonists. Drugs that inhibit the reuptake of serotonin, the so-called selective serotonin reuptake inhibitors, or SSRIs, are often used to treat depression, anxiety, and obsessive-compulsive disorder. These drugs include fluoxetine, Prozac, clomipramine, anafranil, and sertraline, Zoloft.
Thank you for your time.